Welcome to Biology, the scientific study of life. This figure represents abscission of a maple leaf. Abscission is controlled by a change in the balance of ethylene and oxygen. The abscission layer can be seen here as a vertical land at the base of petiole. After the leaf falls, a protective layer of cork becomes the leaf scar that helps prevent pathogens from invading the plants. Notice this figure represents ethylene signal. Transduction mutants can be distinguished by their different responses to experimental treatments. Notice that a constitutive triple response. Notice also control ethylene added ethylene synthesis inhibitor, ethylene overproducing, ethylene insensitive. Notice that, and white type. Another figure represents ethylene triple response arabidopsis mutants. Notice A, picture number A of this figure. An ethylene insensitive mutant fails to undergo the triple response in the presence of ethylene. Notice closer look. Notice also B, a constitutive triple response. A mutant undergoes the triple response even in the absence of ethylene. Notice. Notice here this figure represents a precocious germination of mutant. Coleoptile, notice this one represents coleoptile. Precocious germination of mutant maize seeds. Abscissic acid induces dormancy in seeds when its action is blocked, in this case by a mutation affecting an abscissic acid regulated transcription factor. Precocious germination results as shown in this figure. Notice this is inquiry. How does ethylene concentration affect the triple response in seedlings? Experiment. Germinating pea seedlings were placed in the dark and exposed to varying ethylene concentrations. Their growth was compared with that of an untreated seedling. Result. All the treated seedlings exhibited the triple responses. The response was greater than increased concentration. Conclusion. Notice that the ethylene concentration parts per million. Conclusion. Ethylene induces the triple response in pea seedlings with increased ethylene concentration causing increased response. Again with gibberellins mobilize nutrients during the germination of a grain. Seeds. Notice the differences or notice the steps. Notice alluron, endosperm, and water contains of green seeds, and scutellum, cotyledon. Notice step 1. After a seed embeds water, the embryo releases gibberellin GA, which sends a signal to the alluron, the thin outer layer of the endosperm. 3. The allurin responds to GA by synthesizing and secreting digestive enzymes that hydrolyze nutrients stored in the endosperm. One example is amylase, which hydrolyzes starch. A similar enzyme in our slavia helps in digesting bread and other starchy foods. Notice also 3 sugars and other nutrients absorbed from the endosperm by the scutellum cotyledon are consumed during growth of the embryo into a seedling. Back to the effect of gibberellin treatment on Thambisan seedless grapes. The grapes bunch on the left is an untreated control. The bunch on the right is growing from a vine that was sprayed with gibberellin during fruit development. Notice the size, the differences also the size. Gibberellins, a century ago, farmers in Asia noticed that some rice seedlings in their paddies grow so tall and spindly that they toppled over before they could mature and flower. In 1926, Iwito Kuroso, a Japanese plant pathologist, discovered that a fungus of the genus Gibberella caused this foolish seedling disease between two brackets. By the 1930, Japanese scientists had determined that the fungus produced hyperlongation of rice stems by secreting a chemical which was given the name gibberellin. In the 1950s, researchers discovered that plants also produce gibberellins in the past 50 years. Scientists have identified more than 100 different gibberellins that occur naturally in plants, although a much smaller number occur in each plant species. Foolish rice seedlings, it seems, suffer from an overdose for gibberellins normally found in plants in lower concentrations. Gibberellins have a variety of effects such as stem elongation, fruit growth, and seed germination. Stem elongation, roots, and young leaves are major sites of gibberellin production. Gibberellins stimulate growth of both leaves and stems, but have little effect on root growth. In stems, gibberellins stimulate cell elongation and cell division. Like auxin, gibberellins cause cell wall loosening, but not by acidifying the cell wall. One hypothesis proposes that gibberellins stimulate cell wall loosening enzymes that facilitate the penetration of expansion proteins into the cell wall. Thus, in a growing stem, auxin, and by acidifying the cell wall and activating expansions, and gibberellins by facilitating the penetration of expansions act in concert and promote elongation. The effects of gibberellins enhancing stem elongation are evident when certain dwarf mutant varieties of plants are treated with gibberellins. For instance, some dwarf pea plants grow to normal height if treated with gibberellins. If gibberellins were apparently, these plants are already producing an optimal dose of the hormone. The most dramatic example of gibberellins induced stem elongation is blotting the rapid growth of the floral stalk in their vegetative state. Some plants, such as cabbage, develop in rosette form low of the ground with very short internodes. As the plant switches to reproductive growth, surge of gibberellins induces internodes to elongate rapidly, elevating floral buds that develop at stem types or at stem tips. Fruit growth in many plants, both auxins and gibberellins, must be present for fruits to set. The most important commercial application of gibberellins is in the spraying of tmkin, as shown here, seedless grapes. Notice that. The hormone makes the individual grapes grow larger, a trait prized by the consumer. The gibberellin sprays also make the internuts of the grape bunch elongate, allowing more space for the individual grapes. This increase in space by enhancing air circulation between the grapes also makes it harder for yeast and other microorganisms to infect the fruits.